All right, today we are diving deep into one of the most fascinating and, frankly, powerful tools in all of modern biology, the embryonic stem cell. We're talking about a single cell that holds the potential to build an entire human body. It's a concept that could completely revolutionize how we think about medicine and healing. So let's get right into what makes these tiny cells so incredibly special.
Just think about this for a second. What if we could repair a heart after a heart attack, or maybe even reverse paralysis, by growing brand new, perfectly matched replacement parts from our own biological blueprint? Now, this might sound like something straight out of science fiction, but it's not. It's the very real promise of a field that's built on these amazing master cells. And that promise, that big idea, it has a name. It's called tissue engineering. This is the whole reason there's so much excitement, and it's the engine driving all the research we're about to talk about.
So, at its heart, tissue engineering is really all about building new body parts in the lab. You can think of it like a kind of biological construction project. You've got your cells, which are like the bricks, then you have special materials that act as the scaffolding to give them shape, and finally, you have chemical signals, or growth factors, that are like the foreman shouting instructions telling the cells what to do.
But here's the catch. To build anything substantial, you need a massive, almost endless supply of bricks. And that's where we hit a wall. Most of our regular body cells, the ones scientists call somatic cells, they have a kind of built-in expiration date. They can only divide so many times and then they just stop. So, to build whole tissues, we need a source of cells that basically never gets old.
And this is where stem cells make their grand entrance. They are nature's elegant solution to this problem. They are the ultimate biological toolkit.
You see, stem cells are basically nature's blank slates. They have two really unique superpowers. First, they can make endless copies of themselves. That's called proliferation. And second, they have this amazing ability to transform or differentiate into pretty much any specialized cell you can think of. A nerve cell, a heart cell, a skin cell, you name it. But, as we're about to find out, not all stem cells are created equal.
So, at the very top of the power hierarchy, you have the totipotent cell. Think of a newly fertilized egg, a zygote. This single cell has total potential. It can build the entire organism. We're talking the baby itself, plus all the support structures like the placenta and the amniotic sac. It is the founder, the CEO, and the entire board of directors of a brand new biological company.
Now, take one step down the ladder and you get to pluripotent cells. These have many potentials. They can become any of the 200+ cell types in the actual body, but they can't make those extra support structures like the placenta. So, if the totipotent cell is the company founder, the pluripotent cell is like the rock star chief operating officer who can run any department from marketing to engineering but doesn't build a corporate headquarters. And this is exactly where our hero, the embryonic stem cell, fits in. And finally, we have multipotent cells. Now, these are much more specialized, and we actually have them in our adult bodies right now. A blood stem cell, for instance, is a workhorse. It can create all the different kinds of red and white blood cells, but it can't, decide to become a brain cell. It's like a department head, an absolute expert in its field, but it stays in its lane.
So this table really lays out that whole hierarchy of power for us. You've got totipotent at the top, the cell that can build everything, all the way down to the more specialized multipotent cells. Our focus for today is right there in that sweet spot in the middle, the incredible, mind-boggling versatility of the pluripotent cell. Okay, so let's zoom in on our main character. Where do these incredibly powerful embryonic stem cells actually come from? What's their origin story? Well, just a few days after fertilization, an embryo grows into a structure that looks like a hollow ball. It's called a blastocyst. And tucked away inside is this tiny, precious clump of cells known as the inner cell mass, or the ICM. These right here are the pluripotent stem cells. If they're left alone in the body, they're the ones that would go on to form the entire baby. Now, the journey to actually being able to work with these cells, well, it was a long one. The whole idea of a stem cell really started to take shape back in the 60s. But it wasn't until 1981 that scientists finally figured out how to isolate and grow them from mice. And then the massive breakthrough came in 1998 when two separate teams, in a huge scientific milestone, did the same thing with human cells. And that just opened up a whole new universe of possibilities. But here's the thing. You can't just grow these cells anywhere. They are incredibly sensitive. They need a super specialized, meticulously controlled environment. We're talking about a 5-star hotel for cells, with round-the-clock room service. Yeah, this is absolutely not a set-it-and-forget-it kind of situation. It's a delicate, hands-on, 7-days-a-week job.
It takes incredible patience and precision, because every single day you have to give them fresh food to keep them alive, and this part is critical, to keep them in that powerful, undifferentiated, pluripotent state. And to give you an idea of the kind of control we're talking about, the air in these special labs has to be phenomenally, unbelievably clean. We're talking fewer than 10 microscopic particles per cubic foot of air. That is way cleaner than a hospital operating room. That level of sterility is absolutely non-negotiable to protect these precious cells from any kind of contamination. So how do scientists actually get these cells out? Well, it's this incredibly delicate, multi-step process. First, they start with a blastocyst, which usually comes from IVF clinics with full informed consent. Then, using microscopic tools, they carefully dissect out that tiny inner cell mass. From there, they place those cells onto a special supportive layer of what are called feeder cells. Over time, the stem cells that survive start to form these little colonies, which are then carefully picked, moved to a new dish, and expanded into a stable, endlessly replicating cell line. And that food I mentioned, it's a very specific, very complex cocktail. It starts with a nutrient-rich base, and then they add things like glutamine for energy, amino acids to build proteins, antioxidants to protect the cells, but the most important ingredient is a growth factor, like beta-FGF. That's the chemical signal that is constantly screaming at the cells, "Stay pluripotent, don't change, just keep growing."
Okay, so growing the cells is one thing, but that's really only half the battle. Once a lab establishes a new cell line, they have to prove that it's the real deal. The cells have to run this gauntlet of super rigorous tests to confirm, without a doubt, their identity and their power.
The very first test is an identity check. Scientists have to look for very specific molecular signatures that are unique to pluripotent stem cells. It's basically like checking the cell's internal ID card to make sure it's legit. And this molecular ID card has a few key features they look for. Deep inside the cell, they're searching for master regulator proteins like OCT4 and SOX2. These are the genes that command the cell to stay pluripotent. Then, on the cell's outer surface, they look for a different set of markers that act like a badge, confirming to the outside world that this cell is in its powerful, undifferentiated state. All right, next up is the stability test. Because these cells divide over and over and over again, there's always a risk that genetic mistakes can creep in. So scientists have to constantly check to make sure the cell's genetic blueprint, its chromosomes, remain totally stable and correct. And they have two main tools for this job. Karyotyping literally creates a picture of all the chromosomes lined up so you can visually check for any big abnormalities, like a missing chromosome or a chunk in the wrong place. Then they do something called STR analysis, which creates a unique DNA fingerprint for that specific cell line. This is crucial for verifying its identity and making sure there are no mix-ups in the lab. But even after passing the ID check and the stability test, there is one final, definitive exam that a cell line must pass to prove it is truly pluripotent. This is the big one.
It's called the teratoma assay, and it is the undisputed gold standard in the field. Scientists take the stem cells and inject them into a special mouse that won't reject them. Now, if the cells are truly pluripotent, they'll form a benign, kind of jumbled up tumor called a teratoma. And here is the absolute key. When scientists look at that tumor under a microscope, they must find tissues from all three of the original embryonic germ layers, things like nerve cells, cartilage, and gut cells, all mixed together. That is the ultimate, definitive proof that these cells really did have the potential to become anything.
So after running this intense gauntlet, we have them. Verified, stable, powerful master cells that literally hold the blueprint for life. The science has proven that we can isolate them, we can grow them, and we can confirm their power. But that's really just the beginning of the story. The next great chapter, the real challenge now, is learning how to perfectly command them, how to tell them exactly what to become and when to become it. So as we stand here, on the edge of a potential medical revolution, the biggest question isn't just what these cells can do, but what we can learn to do with them.